Right, so we have basically finished Key Area 2. We do have to tread back through some stuff. We'll probably have to tread back through all of it after the summer anyway. But generally speaking, from the work I got back, I have been very, very impressed and heartwarmed to see that we are grasping this stuff pretty well. I'm going to go through what the thinking is, what the answers are for each of the questions. We're going to take a look at what the marking scheme is for the SQA. Most of you have actually been hitting the wording as they would like it, so that's actually a pretty good first step. Generally speaking, pretty happy. So our first question was this multiple choice one, and we've got a diagram and a transmembrane protein that's stretching in and out of the membrane, so it's crisscrossing the membrane at different points. The big thing that you have to remember is that those tails are non-polar, and that region will want to interact with non-polar or hydrophobic amino acid R groups, and that is all about really the distribution of electrons, whether or not those electrons are distributed evenly or unevenly. So arginine there is at the head section of the phospholipid, and so you're expecting, because that head section has a charge, it's hydrophilic, that arginine would also have a charge and it would be hydrophilic. You would expect these ones that are in the tail section, or actually in the membrane, to be non-polar and hydrophobic. So that's really what we're dealing with here. So if we look down at the rows, arginine being polar, that does seem to fit. So arginine being polar, that fits with what we've got. That head section will be polar. Polar and polar, that makes sense. So alanine being hydrophobic, now that is sitting right in the membrane. These sections here are going to be hydrophobic. That means the alanine itself will be hydrophobic as well. The same argument for leucine, and serine is on the head section, which will be polar, so A is our answer. And that really, this question here was a little bit of a help in terms of the essay question as well, because the essay question was really asking you a little bit about this as well. Now, this would be an integral protein that is well and truly embedded in the phospholipid bilayer. It wouldn't be a peripheral one, which would just have a small interaction. This is pretty well embedded in there. Right. 2, I'm looking at this and I'm immediately thinking it's probably a little bit of problem solving. So, bovine insulin is a soluble protein with an isoelectric point of 5.4. Now, this is actually a pretty interesting little bit. It's basically determining whether or not something has a charge based on the concentration of H-positive ions in a solution, and that will essentially change its charge, because you've got the weak acids in a dynamic equilibrium with the solution, and where we've got the isoelectric point, that means we will have no charge on our protein or our amino acid. We'd usually be looking at an amino acid, I would have thought, for that. Makes it a bit simpler. Protein's probably a bit too complex. Which of the following graphs represents the level of precipitate formed as the pH of bovine insulin solution is changed? Now, this is pretty interesting because the precipitate is going to be created from the thing becoming non-polar and falling out of solution. So the mass of precipitate as we get to 5.4, so as we get to that middle section, should increase, because as you get to the isoelectric point, it's going to lose its charge and it's going to become less soluble. So the answer for 2 should be C. That should be C for 2. And that was, to me, that was a pretty tricky question. Quite frankly, that's getting on for using all your knowledge about chemistry and biology at that point. That's a pretty nasty question. So the three-dimensional structure here, that little section that is X should be an alpha helix, and that will be hydrogen bonding that stabilizes that structure. Remember, that is when you're talking about the primary structure, you'd be talking about the peptide bonds in the order. When you're talking about the secondary structure, you're really talking about hydrogen bonding, and you're not really worried about those peptide bonds. They're already in place. So 3 should be D. That should be D because we've got an alpha helix and it's hydrogen bonding that's stabilizing that. Obviously, we've covered that those alpha helixes are due not just to hydrogen bonding, that there'll be a whole host of bonding that's creating that, but the main thing that we're talking about is hydrogen bonding, and that's simply due to the fact that we've got lots and lots of water. This is a hydrated molecule. It's a molecule that is the shape that it is because it's surrounded by water molecules. Okay, 4, nice and diagrammatic. The diagram shows some interactions between amino acid R groups in a polypeptide chain. Which letter shows hydrophobic interaction? So if we've got hydrophobic, we have no charge. So A, that is showing the slightly positively charged hydrogen being attracted to the negative charge on an oxygen. So that's interesting. That's probably not quite an ionic bond, but it's probably quite close to an ionic bond. That would be a hydrogen bond in A. So that would not be a hydrophobic. That would be a polar interaction or a hydrophilic interaction. That is not hydrophobic. B, they're often called aliphatic chains or hydrocarbon chains. These are non-polar, and they are essentially pushed together not because there's necessarily an interaction between them, but because the water is trying to exclude them, and it's the exclusionary force of the water that pushes those together. That exclusion of water, or that exclusionary force from the water, is that hydrophobic interaction, and that is B. So 4 is definitely B. D shows you an ionic bond, and C is a disulfide bridge, which would be a covalent bond. So, yeah, B is that one. So, the proteome is larger than the number of genes in the genome of an organism because... So, what we're really talking about here is you can kick out a lot more proteins than you have actual numbers of genes, and there's a few reasons for that. Alternate RNA splicing, and you've also got post-translational modification that can give you different functioning proteins. So, A, not all genes are expressed as proteins in a particular cell. That is true, but it doesn't answer the question. So that is very, very true, but it does not answer the question. B, post-translational modifications generate multiple RNAs from a single gene. Now, this is an interesting option because they have thrown in post-translational modification, but then they have stated that that is what generates multiple RNAs from a single gene, and that is not true. That is C, alternate RNA splicing, or alternate splicing generates multiple RNAs from a single gene. So B is really chucking in all the things in the wrong fashion. C has one thing in the correct fashion, so the answer for 5 is C, and that was the multiple choice section. All the marker's not even really going to do this, but it'll be a machine that does it, and that's what you've got in the marking scheme. This is obviously a marking scheme that I've piled together from those questions, but that is our multiple choice section. Okay, so once you've done that multiple choice section, you're going to come on to the short written answer section. This bit is going to give you usually some pretty tough questions, and they're going to build in some problem solving, some wider knowledge. You will not have come across in the course all the stuff that's spoken about, because they will give you essentially a story, and they will be asking you to use your knowledge of the course to answer questions on that little story. So, our one short answer question that we had in the test was about sickle cell anemia. This is an inherited blood disorder that reduces the ability of red blood cells to transport oxygen around the body by changing the structure of hemoglobin. In sickle cell anemia, the primary structure of hemoglobin subunit is altered. The amino acid glutamic acid is substituted by the amino acid valine. The structure of glutamic acid and valine are shown here. Now, you want to take a minute to just look at the structures and look at the differences. So, the one thing is they've highlighted the R group and the amino acids in a little bit. It's odd to see an amino acid drawn like this. It's actually not the way that I would draw an amino acid. I would have the R group sticking up. I would have the acid section on one side and the amino section on the other side. That is not shown here. You've got this right angled. There's nothing wrong with it. It's perfectly reasonable, but it's not how it's usually drawn. However, the R group's really telling you the main thing that we've got here. So we've got this charged R group on the glutamic acid. A hydrogen's essentially plopped off and left this negatively charged oxygen. So that is going to be very hydrophilic. It is charged and it's going to react very differently than something that is non-polar and hydrophobic, which is what we're seeing in the valine. So these almost have the opposite properties, just to start us off with. So state the class of amino acids to which valine belongs. So as we were saying there, you could either say non-polar or you could go for hydrophobic. So you could say that that's hydrophobic or non-polar. Either one of those two is fine. And that's what they actually have in the answer scheme. So either of those two responses is fine. Identify one type of secondary structure shown in the hemoglobin molecule in the figure below. This is what we actually saw on PiMol. Hemoglobin is quite an important thing. They talk about it quite a lot. So when we get a chance to do some proper lessons, it's certainly something that we're going to take a look at. We're going to use it as an example quite a few times throughout the course. So, one really obvious one that I can see is the alpha helixes. You could get away with turns. The turns are really the connections between the alpha helixes or beta pleated sheets. So you could either say turns, which are these little bits here hanging out, or you could have said the alpha helixes, and I think both of those are represented in the answer scheme, so you get 1 mark for that. Right. Graph questions. Explain the term cooperativity in relation to oxygen binding to hemoglobin. Now, that is where you've got a quaternary structure and you've got multiple polypeptides that can be altered. As oxygen binds to one binding site, one heme group, it increases the affinity of the other heme groups for oxygen. So it means that as one oxygen is bound, the binding affinity for the other heme groups for oxygen increases. A few of you said this in lots of different ways. I think, honestly, most of them would have got it. The answer they're looking for is makes the binding of other oxygen more likely. Correct reference to affinity, change for binding, release of oxygen. Most people that got the mark had something in that line of thinking. That was really well done. It was a pretty tough question. Alright, if you've had me as a teacher before, you will have heard me moaning about quoting from the graph, potentially even using highlighters. It's a tactic stolen from Dr. Hayhurst, which is fantastic. This is worth 2 marks, so you've really got to make 2 statements. If you only make 1 statement, then you are only going to get 1 mark, and that was the case for a few people. Some people knocked out the park, got 2 marks, even made all 3 points that are available to make. So, if we quote from the graph, in fact, I should probably read this bit first. The graph below shows oxygen saturation of hemoglobin at different oxygen pressures for an individual with normal hemoglobin and for another individual with sickle cell hemoglobin. So our sickle cell is our dashed line, our normal hemoglobin is our unbroken line. We've got oxygen saturation in percentage here going up from 0 to 100%, and we've got oxygen pressure in units. I mean, if they haven't given it in bars or anything like they could have done, but they've made it simpler by just saying units and giving you 0 to 100. Interestingly, that's probably making it easier than some of the higher graphs that you've come across. Right, so use the graph to compare oxygen saturation of normal and sickle cell. So if I was to quote from the graph, then what I would be saying is between oxygen pressure 0, and I would have oxygen pressure units at 0 and oxygen pressure 16 units, both normal and sickle cell hemoglobin are the same. So the oxygen saturation for both normal and sickle cell hemoglobin between 0 and 15 or 16, oxygen pressure is the same. Between 15 or 16 oxygen pressure units and 100, the oxygen saturation is lower in sickle cell hemoglobin than normal hemoglobin. And at oxygen saturation of 95% at oxygen pressure of 100 units, for the normal hemoglobin, you only get, and that's 50% oxygen saturation at the same pressure for sickle cell hemoglobin. So really, it's probably quite difficult for you to run with me saying that verbally, but I am quoting from different sections of the graph, and then I'm highlighting a specific section that's different. So if we look at the answer scheme, you get 2 marks. So you get 1 mark for saying at low pressure below 15 or 20 there is no difference. So that's quoting from the graph that between 0 and 15 there is no difference between normal hemoglobin and sickle cell hemoglobin. And you see this really in the pathology of this disease, where unless people exert themselves, there's actually not a huge difference in their ability to function. It's only when they exert themselves that the pathology really presents itself. Then you get 1 mark. You can't pick up both these marks. You either get one or other. comparison of the saturation at high levels, so that's me comparing that 50%, or just stating that at high pressures, the normal saturation is greater than the sickle cell saturation. That's how you get the 2 marks for that one. Now, this question was fantastic. It was really quite brutal. So, molecules of sickle cell hemoglobin clump together, preventing access to oxygen binding sites. Whoa! And that's because the glutamic acid has been swapped for the valine. So if we go back up and we take a look at these two, what they're saying is the heme site for a normal hemoglobin has this glutamic acid, whereas valine has been swapped in there in the sickle cell. Now, if you've got glutamic acid and you've got that negative charge, it's going to be repulsed by the other negative charges within that heme group at the oxygen binding site. The R group for the valine is non-polar, and it would not repel other negative charges, and so it would end up clumping inwards. So you had to really state something about the R groups being different and the presence of that negative charge being able to repel other negative charges and keep the heme group open. So if we take a look at that, valine has no charge in the R group, so molecules don't repel one another. Hydrophobic interactions occur between R groups of valines, causing clumping. Now, there's a little bit of a problem in terms of how they've asked that question, quite honestly, because what they should have said, which you don't know, is that there is other glutamic acids in that heme group that all make up that site. So actually that question to me is a little bit harder than it should be. They should have made it clear that there's multiple glutamic acids all in that heme site, and that the swapping of that glutamic acid for valine causes the heme site to collapse in on itself. That is not made clear throughout this, and it's certainly not in the course documents. So it's a really hard question. If you had stated something about the R groups being different, that was getting you on to getting that mark, really. And saying that you wouldn't get that repulsive force from the other R groups being negatively charged would get you. Pretty hard one. But overall, people dealt with that pretty well. I was pretty happy with it. It was a good question, and the graph was done pretty well. Just remember to quote directly from the graph. The graph questions should be gimmies, because you're just reading from the graph. Take your time. We'll go over graphs properly. We'll talk a lot about how we get the most from it, how we use highlighters, especially on the 3-sided graphs, and how we extract the most marks from it. So that was our short answer section. Right, the dreaded essay questions. The reason these are difficult is because your thinking isn't structured. There's nothing really there to help you determine where you're going, what they're asking. How you interpret the question is extremely important, and it really gets to the heart of some of the really big issues that you're going to have, is that they could ask you something that you know the answer to, but the way in which they have asked it is technically quite difficult. So the question that I'd given you was, discuss the role of amino acid R groups in the following. So the first one's only worth 3 marks, and it's interesting what you get those 3 marks for. So first of all, discuss the role of amino acid R groups in these two things. The first thing that I would do is I would make a list of the stuff I know about R groups on a separate sheet of paper. So, I wonder if this will work. That's probably a terrible idea. So, what do I actually know about R groups? What am I thinking about when it comes to R groups? Well, one of the two basic things that I'll be thinking about is and there's my starting point. So I've got R groups that are hydrophobic, I've got R groups that are hydrophilic. So this is me starting my thought process. And during an exam, especially when you're coming to the end, you're tired, you've done quite a lot, you've been in there for possibly 2 hours already, it is important to just take a minute and think through what you're gonna think about. So now I've got my 2 main points that I want to talk about for R groups, and I really want to talk about how R groups either make something hydrophobic or something hydrophilic. So the first question is asking me to say how these R groups determine the tertiary structure in proteins. So the first thing that I want to do is I want to define what that tertiary structure is. So that is the overall 3D shape of the polypeptide chain. Now, that overall shape is determined by the order of the amino acids in the primary structure. So the order of amino acids in the primary So that order will determine the overall shape. And what I want to do is I want to bring this back into the R groups and talk about the R groups. They have asked you specifically to talk about those R groups, so you're going to have to talk specifically about those R groups. So the order of R groups will create interactions that cause folding. These main folds or motifs down. So, now I've basically set up, my first line of thought was hydrophobic, hydrophilic, and I've got to talk about that still. I've defined what the tertiary structure is, so it's the overall 3-dimensional shape of that polypeptide chain. It is that shape because of the primary structure. Those R groups create those secondary structures which then fold and contort into the 3D tertiary structure. I have then probably got to say Oh, go sneeze. Nope, I'm fine. Depending on the R groups, different types of bonding will occur, and list them come off the page. These bonds will hold the polypeptide chain in a three-dimensional shape. Right. In essence, what I have said there is my answer to that question. So, hydrophobic and hydrophilic, I haven't really spoken too much about that, but I've given the hydrogen bonding, the disulfide, but we'll see. I've given the definition of that tertiary structure. I've spoken about how the order of the R groups is important. So, if we check, this is our bit. It says max marks 9. It's only 3 for section 1 and it's 6 for section 2. So tertiary structure is a folded polypeptide three-dimensional shape. The shape, structure, conformation, folding is determined by the order of amino acids, so that primary structure. So you had to say that. That'll give you 1 mark for the definition. Saying the order of the R groups or the primary structure would have got you 1. 2 R groups named from either polar, hydrophobic. So I haven't done that. I didn't actually go into that kind of detail. I just said the types of bonding that would occur. So what I might want to say is, I've moved that full stop. The R groups can be polar, non-polar, charged, so that could be positive or negative. That would be, acid, and that would be basic. So that little bit adds in. My dog's currently going a bit mad. So we've covered that, and then name the types of interactions. Only 1 from those would have been enough. Did I miss any out? Hydrophobic interactions. Cheeky. And so that's really how I would construct this up there. So that was part 1. Part 2, I'm going to be thinking about those hydrophobic and hydrophilic interactions even more, probably. So that would be how I would build it up. I would start by writing down what I know about the R groups. I'd constantly refer back to the R groups. You've got to remind yourself about what's happening in these essay questions. So you've got to remind yourself, what do you know about the protein structure? And they're going up to the tertiary structure, and so they are really wanting to know how we get from that primary order of amino acids to the three-dimensional contorted shape of that protein, and how those R groups, being polar, non-polar, charged, positive or negative, create bonding, and that bonding will cause that contortion in shape. So that would get you the marks. We'll do essay questions over and over and over again. I don't want that. I want to close that. We will do essay questions a lot. We'll do one of these at the end of every key area, and we'll come back to them as well. We're not going to leave key areas once we've done them. We're going to keep coming back to them, keep talking about them, keep going over it. So, I've got a fair bit of space. So, 2 was influencing the location of proteins in the cell. Now, when I was marking this, I'd actually forgotten to think about myself when I saw that question, because I was like, that's worth 6 marks. That's quite a lot. So really, my first port of call is to say that the tails of the when anybody writes respectively, it means that how the sentence goes is how it's meant. So the head section is charged and the cytoplasm is polar because it's got it's made up of water molecules majoritively. You could even make the argument that because it's got so much dissolved ions in it that it's charged as well, but we'll stick with that without getting too deep into that. So I have really got to state that the R groups, because remember, this is still in reference to the R groups, so a protein with non oh oh my hay fever's really getting to me today. Okay, so a protein with R groups will cluster in the non-polar tail section a membrane if those a protein with non-polar R groups will cluster in the non-polar tail section of a membrane. Okay, so now I'm thinking about membrane proteins. And if you remember, we've got transmembrane proteins and I spell that right? Peripheral. Let's check. I'll be in the marking scheme. I should probably have split that up differently as well. Okay. I want to split that up differently. So in the marking scheme, what they're saying is we've got integral and peripheral. And there was me mixing that up, that's not very good. So, the integral proteins are going to interact strongly with the membrane. So, I probably want to keep the sentence structure the same. Integral proteins bind loosely, weakly. I really want to say for a short period of time. Oh. Oh my god. Oh. Okay, right. So, what I've got down there is we have split our two types of membrane proteins into integral and peripheral proteins. The integral proteins are really part of the membrane in essence. They are much like that question there. They're really going through the membrane. They'll probably go through it several times. Their interaction in that section of the membrane is going to be pretty strong. They might have bits that come out into the extracellular matrix, or they've got some on the cytoplasm side into the cystole. just depends on the protein. And those integral proteins bind indefinitely, so they'll be there for a very long time. They've got a strong interaction. These can be transmembrane proteins, channels, pores, and pumps. The R, and I want to bring it back to the R group because that's really the question. Oops. Are important as they will be whether a protein can polar region of the membrane as the R groups will need to be non-polar as well. Now, if you wanted to be really fancy and be almost cheeky, but not really, we've got a question here. Short answer question, which gives us the name of an amino acid that's non-polar and the name of an amino acid that has a charged group on it. So you could even use that as a for example. For example, the R group on valine is non-polar and so could interact within the tail. Interact with. Okay, so this was worth 6 marks. The marking scheme, and we'll speak about which ones we've got at the moment. We've not even really spoken about solubility. If a within the could even talk about those globular proteins in, I think that was in the marking scheme as well. Oh. Yep, we've done that. So we spoke about the membrane and the tails being hydrophobic and the head being hydrophilic. I think we did, didn't we? Oh. Yes, we did. So we did that one for the membrane. We spoke about this. We now have spoken about the soluble proteins being found in the cytoplasm. I think I at least have a really bad picture of solubility in my head from learning this in chemistry when it would have been standard grade level. And it's really from the idea of salt. When you put a salt crystal into water, it'll dissolve because it's soluble, and it'll break the crystal apart. Solubility doesn't necessarily mean that the structure is broken apart. It simply means that it will be miscible within the water, so it will mix into it. I think that might even be the wrong use of solubility. I'm not going to argue with it at the moment. But basically, the protein, if it's soluble, it will not form a precipitate in water. You're not going to find it forming a solid at the bottom of the water. It will be mixed in to the water, but it doesn't mean that it's going to break apart or anything like that. It just means that it's going to be floating around there with water molecules surrounding it. It's not going to be pushed out like oil would be, or some other insoluble substance. So that's something that we probably want to speak a little bit about. That word, I don't actually think that's being used correctly. I will actually take a look at that, and we'll speak about that later. Solubility, the wording for solubility can be a little bit tricky, so it's probably worth coming back to that. Right, moving on. Blah blah blah. We did that. We spoke about proteins as integral and peripheral, and we needed both terms. We got those. Some integral proteins are transmembrane. One of the following. Integral proteins, the idea of hydrophobic R groups interacting with that's the main bit really that I've spoken about and I've fixated on a little bit. That's another important point. When I did this the first time, I really fixated on that bit, and I just spoke about these two bits ad nauseum when I did the questions the first time round, and I missed out quite a few. I didn't even speak about solubility. Completely forgot about it. Completely forgot about it the second time there as well. I spoke about I spoke about these bits. So what would I have got? One, two, three. I'd only got half marks really, because I'd forgotten about integral and peripheral proteins. I spoke about transmembrane proteins maybe 4 out of 6. So I didn't by any stretch of the imagination knock that out the park. I'd completely forgotten about integral and peripheral. And speaking about that, I'd forgotten about talking about solubility of proteins in the cytoplasm. Just because of the way the question was, really, it was quite flat as far as the question goes. But that's how I would build it up. That's the answers to it. We're going to need a lot of practice at the essay questions. We will get there. And those are the two answers that I would give. It's how I would build it up. I'd go from what do I know, I'd write that down, and I'd start constructing my thinking around it and keep adding to it. You're not gonna lose marks for adding more. Right, before I have another sneezing fit, I am gonna say goodbye. Hopefully that was useful. I am a little bit behind again, so I will try and get us started on Key Area 3 this week. So and good luck.